Thank you for that introduction. I'd like to talk about what we've achieved in the last 3 years or so in terms of going towards a cure for HIV. Obviously, we're only there yet for one person, but there were several people that were thought to be cured for a short period of time. So I'll talk about what we've learned from all these cases. As an overview, I'm just going to talk about what is the HIV reservoir, how do we measure it, and then the concept of antiretroviral intensification, adding new drugs to an already succeeding regimen. Prevention of reservoir establishment, so I'll expand more upon that. Stem cell transplants, latency reversing agents, gene editing, therapeutic vaccines, just to touch on. And then broadly neutralizing antibodies, which Dr. Ehler has been waiting for, and radioimmunotherapy. So let's get started at talking about the reservoir. So there are two ways to think about the reservoir. The first is the cellular reservoirs. So we all know that HIV basically infects CD4 positive T cells, usually through the CCR5 receptor early on and the CXCR4 receptor later on in infection. And there are some memory T cells that become dormant, and then these become the core part of the reservoir, which is difficult to eradicate and which makes HIV difficult to cure. There are other types of cellular reservoirs like dendritic cells, macrophages, mostly gut-associated lymphatic tissue. That's a big part of the reservoir. And then we're starting to just find out more about anatomical reservoirs. And you can see the different font here. I've added kind of all the new things that have come out since this article or this slide in 2008. So we've known that the central nervous system can be part of the anatomical reservoir, and a lot of our first-line antiretroviral drugs do not penetrate the CNS very well. Cardiac tissue, epithelial and endothelial cells have been shown. Adipose tissue, which is very interesting. Difficult to eradicate that, as we know. And then the GI tract, which I refer to. Kidneys as well. So the kidneys have been found to be an independent reservoir in HIV, and the testes. And what some of these, like the testes and central nervous system, share is like a blood-brain type barrier or a blood tissue barrier. And so it's sometimes difficult for antiretroviral drugs to penetrate through these barriers. I'll talk a little bit about the different types of T cells just in brief. This could be like a whole lecture in and of itself. But basically you start off with naive CD4 T cells. Then those turn into memory stem cells. Then central memory T cells. And as you go towards the left side, these are the most long-lived cells here. And these are shorter-lived cells over here, like the terminally differentiated T cell. Interestingly, we found through some studies that if you get antiretroviral therapy earlier, it kind of changes the composition of the reservoir. So I'll talk about more of that in just a moment. And so basically the way we treat HIV can kind of change the reservoir. And as you could see, as you go from left to right, you get kind of more of those long-lived memory cells, the blue and dark and light blue cells, and less of those short-lived T cells. So the reservoir becomes this core of difficult to eradicate cells. There are some studies in which they looked at kind of the decay of the reservoir, viral decay they call it, and basically some studies have shown that it might take about 60 years of antiretroviral therapy to get rid of the reservoir completely, so most people don't have that much time. So we really have to figure out other ways to help them out. Measuring the reservoir is really a big challenge. How can we say that somebody is cured from HIV? That's difficult to say. So the first kind of gold standard, I'll just talk about some of the different measures that I'll be referring to in these studies, is the viral outgrowth assay. Basically what they do is they take pherese T cells from the patient or peripheral blood mononuclear cells, so those are mostly lymphocytes. That's how they'll kind of be referred to throughout the talk. And then they take those cells, and this will be the top one I'm talking about here. So they take those cells and then they put them in kind of in vitro, and they take uninfected immunoblasts from HIV negative patients, and then they combine those and basically grow the virus in these immunoblasts. And then they do an ELISA test to one of the P24 antigens to HIV, and then they can kind of measure the reservoir. So you see them a lot of times express in 10 to the 6 cells. So they take a lot more blood than we would require in just a normal viral load. These are very expensive tests, and they're difficult to perform because you need a specific expertise in doing so. But that's kind of the gold standard by which all these other ones are measured. There's also ultrasensitive droplet digital PCR. That's kind of the lower panel that I'm referring to. Basically, they're able to take a tiny sample of blood and to split it into 1,000 or more little droplets. And each of these droplets goes in a well, and they run a little PCR polymerase chain reaction type of reaction in each one, and then you get a positive or negative in each well. So you'll have 1,000 wells, let's say, and you'll have like 3 positives, and then you'll know that the viral load there is 3. So that's kind of an oversimplification, but it gives you an idea of kind of how sensitive these tests are becoming. There's also what's called a single copy assay. It works similarly, but basically it doesn't use that dispersion of droplets, but they're basically able to get down to a single copy per ml of blood. So I'll spare you the details of that, but know that the tests we're using now are much more sophisticated. We also look at, so some of these PCRs can either be done on DNA or RNA. As we know, RNA kind of detects the kind of free-floating HIV in the plasma often, and then DNA will detect it also incorporated into the host genome as well. So it detects free virus inside the host cell, and then the one that's just been incorporated. And then we also look at, some studies have looked at these things called 2 long-term repeat circles. These are basically HIV, kind of a circular HIV that's kind of in the cytoplasm of the cell and kind of getting ready to be assembled. And some people say that these are kind of a marker for active replication. So in some of the cure studies, they kind of look at these to see if there's any active replication going on. This is kind of a controversial topic, whether these are actually long-lived or very short-lived. So I won't really go into the details of that, but know that that's kind of an assay that's out there. So moving on, basically, is more of the same better? That's a good question. So we've looked at, maybe we're just not giving patients enough antiretroviral drugs. There's something called cryptic replication, and basically, even if you can't detect the level of viremia in a patient, let's say it's below your detection level, let's say less than 20, is there still some ongoing replication happening? So they looked at this study. This was presented at CROI back in 2014, I believe. This is called the AirImmune 2 study. And they basically added on to whatever regimen these 28 patients had, and they took them and randomized them into 2 groups. They were also doing like a therapeutic vaccine trial, so half the patients got a therapeutic vaccine, plus the addition of Maraviroc, so an entry inhibitor, as well as raltegravir, which is very appropriate that we just talked about that. So 14 patients got just the addition of those 2, and then 14 patients got the addition of those 2 plus the therapeutic vaccine. And they looked at kind of the HIV DNA, so a measure of the total reservoir, incorporated and unincorporated HIV DNA. And as you can see here from this slide, basically, they don't really show any big change in the reservoir over time. So in these 28 patients, the addition of these 2 drugs did not really eradicate any of the reservoirs, at least over the time period in the study. They've also done similar studies in France in about 22 patients, and that has been a negative study as well. And there are a few other studies that have looked at this as well. So it looks like more is not better, at least with the current drugs that we have. So there's another study that I'd like to talk about. It's called the Visconti study, and they basically had a large cohort of HIV-infected patients, and then there were some patients out of that cohort that they realized were getting HIV treatment and were remaining undetectable even after they stopped treatment. So looks like the average length of somebody to rebound with HIV detectable virus is about 14 days or so. And then some of these patients were basically going for months, up to 89 months without detectable virus, or some with small viral blips. So they studied these 14 patients, and then they compared them to a group of 8 HIV controllers, which we also usually call HIV elite controllers. And they look kind of at the difference between elite controllers and these post-treatment controllers. So post-treatment controllers, basically the definition is you have to, at least for this study, you had to have undetectable viral load, which was less than 400 copies for at least 24 months. And then most of these patients, interestingly, were started on treatment very early in HIV infection, usually within the first 10 weeks of seroconversion. So they had very accurate ways to be able to identify who, when they seroconverted. Let me just see here. Okay. Thank you, Dr. Ehler. Okay. Okay, so we're back. They were on antiretroviral therapy after, testing positive for HIV and kind of being treated in the initial few weeks of HIV for about 3 years on average. And then most of these patients, the 14, were able to have suppressed viral load, 8 completely and 6 with little viral blips. And you can kind of see, hopefully you can see from this slide, each one of these different little graphs is a patient. And here, kind of you can see these blue lines here. These represent the plasma viral loads. And the gray boxes here, for example, the shaded gray areas, indicate when they're on antiretroviral treatment. So you can see that the majority of these 14 remain fairly undetectable even after stopping antiretroviral treatment. So they tried to figure out exactly what was special about these patients. And what was special about them is that when they started HIV treatment early, basically it changed the nature of their viral reservoir. So it shifted the viral reservoir from kind of those longer-lived stem cell and memory T cells, central memory T cells, to these transitional T cells. So there was a higher proportion of shorter-lived cells. So treating HIV earlier is better. So we know that now. And then long-term non-progressors, a different HIV group, are also known to have less central memory T cells composition making up their viral reservoir. So this kind of adds more evidence to that. So I'm sure everybody's heard about the HIV positive baby that was quote-unquote cured. We call this the Mississippi baby. Basically, the mother was HIV positive. She presented in labor with no prenatal care whatsoever. That's great. And then she got no antiretroviral therapy either before or during delivery, because they got the positive test back shortly after delivery. And the interesting thing about this infant is that they started antiretroviral treatment within 30 hours of life. The patient got zidovudine, lamivudine, and nevirapine, and later on the nevirapine was switched later to lopinavir and ritonavir. And basically, the patient, the baby was doing well. They had verified the HIV positivity of this baby. And at 15 months, this is the other unique thing, is first unique thing was the baby was treated early. Second unique thing was that the antiretroviral treatment was stopped because the mother was non-compliant at that time. So they basically observed this baby, and at 24, 26, and 28 months, they tested negative for HIV. So this kind of created headlines, maybe we treated this baby early enough so that it prevented the establishment of reservoirs and cured HIV, or it came to what's called a functional cure. They also did some genetic testing, and it didn't have any of these kind of protective HLA types or CCR types, CCR5 types. Unfortunately, there was some rebound of viremia, so we know that the baby's not cured. She is back on antiretroviral treatment. And there are two other, and I could just mention some of the things in this slide here. Basically, at 24 months, they kind of looked at the viral reservoir, and you could see kind of, this is the number of positive tests. Only one positive test at 26 months came back for virus, and that was in the proviral DNA. So that includes both incorporated and unincorporated DNA into the genome, and some of that is probably not replication competent DNA. So that's one of the limitations of doing PCR tests, is you don't sometimes pick up HIV DNA that's not going to really cause infection. But in this case, there was rebound, and we did learn some things from it, comparing it to other infants that were infected. There was actually a Canadian infant infected and then they started off antiretroviral treatment within the first 24 hours. And then a Milan infant who started off in the first 12 hours. So I'll kind of compare these 3 infants together and kind of tell you what we learned from those studies. So starting off, I highlighted in red kind of what characteristics would be less beneficial and which ones would be more beneficial. So if you look at the onset of antiretroviral therapy, the Milan infant at 12 hours had the shortest time to initiation of therapy. And the time on antiretroviral treatment would also go towards the Canadian and Milan babies. They were both given antiretrovirals for 3 years, and then just 1.5 for the Mississippi baby. The time to HIV suppression was actually in favor of the Mississippi baby. And this may be why that baby was able to go 27 months rather than just less than 1 month without viral rebound. So maybe the time to getting undetectable is important. So that's what we kind of learned from this study, at least. It's a very small, limited sample, but it gives us some idea of what's important. Next, this is actually Timothy Brown. He is the only person that has actually so far been cured of HIV. He's released his image and everything, so everybody knows who he is. He's referred to as the Berlin patient because he was living in Berlin at the time. And at this point, he's been undetectable from HIV for 7 years. So this article came out of the New England Journal. It kind of summarized his case. He was a male, 40 years old, AML M4, and then he was diagnosed with HIV about 10 years previously. He was treated with efavirenz, tenofovir, and emtricitabine leading up to the transplant, and then they stopped antiretroviral treatment for the transplant. He had relatively reasonable viral load, undetectable viral load, and a reasonable CD4 count of 415 before going in. This kind of summarizes here his timeline. So he developed the AML. He had a relapse of AML, so he was receiving chemotherapy in this time period. And then he received a allogeneic hematopoietic stem cell treatment. So that's right around here is his day 0. This represents his plasma viral RNA here, this graph, and this represents his CD4 count here. And on the bottom, you can see what kind of treatments he was getting. So he was getting for a graft versus host prophylaxis, antithymocyte globulin, and cyclosporin, and then mycophenolate mofetil. He also received total body irradiation. This may become important later. So he received a very aggressive conditioning regimen. For conditioning, he also received cytarabine and gemtuzumab, I should mention. And then basically what happened is he developed a relapse. So he had a second stem cell transplant. He was given, and the most important thing, I think, is that he got a delta 32 CCR5 negative homozygous negative transplant. So they basically combed through all the people who he matched with in Europe, and they selected the one that had 2 mutations for the CCR5 receptor, making him relatively resistant to getting HIV in the new donor cells. So this, I think, is probably the most important part of what happened. And then he got, so I mentioned his second stem cell transplant he got from the same individual right here. So they subjected him to a lot of testing. You can see in this kind of green box here all the different groups who tested him, including the NIH, Johns Hopkins, just to name a few. He was at University of California, San Francisco, and San Diego. And he underwent CSF studies with a lumbar puncture. He underwent rectal biopsy twice, basically through sigmoidoscopy and colonoscopy, and all kinds of invasive tests, leukapheresis, a inguinal lymph node biopsy. So they looked in all these various places to see if they could find HIV in him. And this is the result of the testing. Basically, they sent out his samples to many different labs to try to see maybe some labs will come back with positives because they have more sensitive tests or different tests. And you could see that in these samples here, only 2 labs worth could detect plasma HIV RNA, and then some rectal biopsy cells had HIV positive DNA. His viral outgrowth assay, which is, as I said before, the gold standard, was negative. And basically, we can kind of consider that he is cured so far because he has had no rebound of plasma viral load. And that's at 7 years. So they also looked at just a little bit about his immunology as well. Does he still have, antigen to HIV? He still does have antigen response, so he's still HIV positive on tests. But that's actually, indigenicity is decreasing over time. So maybe there is one day in which he will actually show up HIV negative on tests. That remains to be seen. So that's our only real cure so far. And time will tell whether, or not he continues to stay cured. But another interesting thing I should just mention is they looked at the tropism of his CCR5, CXCR4 virus. And he did have a small proportion, about 4% of his CXCR4 virus before undergoing the transplant. So he did have the possibility of having a different type of virus infect him through a means, that would get around the CCR5 mutation. And that so far hasn't happened yet. So that's interesting. But that's one mechanism by which I guess we could foresee him potentially failing in the future. But it looks like he's had 100% donor chimerism, so I don't think that's very likely to happen. So you can break down into 2 big categories, the types of treatments that he got. One is, of course, the myeloablative chemotherapy. So that basically destroyed a lot of the HIV-infected cells already. And then, the process of getting an allogeneic stem cell transplant. There's, of course, a graft versus HIV infected cell effect, just like there's a graft versus tumor effect or whatever. So which one of these components was really the big part that played? So they did actually studies, kind of retrospective analysis. This one is of 10 patients, and you can kind of see the median days post-transplant that they looked at these patients. These are all auto transplants, so they got their own cells back as rescue for HIV lymphoma. And they looked at these patients at about a median of about 2 years later after they received their transplant. And then they were still able to find, you can see here in the red, HIV DNA in all of these patients. So it looks like the myeloablative chemotherapy was not really what cured him, at least as this study would indicate. There are also, I don't know if you've all heard of the Boston patients as well. There are 2 Boston patients that were thought to be cured from HIV just similarly to him, but they've both rebounded with virus as well. But initially, it's interesting to talk about the differences between Berlin and Boston patients, so let's do that now. So basically these 2 patients, one was perinatally infected male. He's Boston patient A, I'll call him. And he had nodular Hodgkin's lymphoma. Basically, he received kind of a reduced intensity conditioning therapy. So that's one difference between him and the Berlin patient. And then he received a mismatch, unrelated donor transplant. And then here you can kind of see up at the top, whenever his viral load was detectable, I kind of outlined it in red. This is the HIV DNA load in his body, so you can see this kind of represents, the area under the curve would represent his HIV reservoir here. It's decreasing here, so they were able to measure that. And then these are days after transplant here. He actually developed really bad graft versus host disease of multiple organs, and as compared to the Berlin patient who developed kind of mild graft versus host disease of the skin. So presumably this patient would have had a very robust, anti-HIV effect because he was having a really high graft versus host disease. Both of these patients, Boston patient A and B, actually had one mutation for the CCR5 gene. So they thought that maybe initially going into this transplant, they were relatively protected from HIV. And then, unfortunately, after what they did is they replaced that CCR5 mutation with a patient's bone marrow that had both wild type, so it had unmutated CCR5 receptors. So Boston patient B, I'll just talk about a little bit. He was a male, sexually acquired HIV in the 80s, and he had a kind of a slow progression, maybe due to his one mutation in the CCR5 gene. He basically had a diffuse large B-cell lymphoma, and then also had Hodgkin's disease after that, and then got an auto transplant and then an allo transplant. So with him, he basically up here did not have any detectable viral load before or after the transplant. You could see his HIV reservoir was pretty big initially, but actually they couldn't detect it. And both him and patient A had remained on antiretroviral therapy, before the transplant, during, and after. And they were both on basically a triplet, tenofovir, emtricitabine, and then efavirenz. So, and they remained on treatment maybe like 2 to 3 years, depending on the patient afterwards. And then they said, let's take them off because we can't really measure any viral reservoirs in these patients. And within 8 months, patient B relapsed, and 12 or 16 weeks later, patient A did. So things that are different between these patients and the Berlin patient, like I mentioned, is the reduced intensity conditioning. So they didn't get as aggressive chemotherapy, and they didn't get the CCR5, homozygous bone marrow. So those are two things that I think were important in his cure. Another kind of, just shifting gears, another line of attack against HIV is this thing that they term shock and kill. Sounds pretty sexy. Sounds like a spy thing, right? So we kind of broadly refer them as latency reversing agents. And then there's some called, basically HIV kind of incorporates into the host genome, and it kind of remains curled up in the chromatin. And histone deacetylase kind of keeps it in there, quiescent, and just kind of waiting to pop out later. So these latency reversing agents like vorinostat, prostratin, and byrinostatin, basically they have been shown to kind of wake up the cells that are infected, the reservoir cells, and they come out and stick their heads out, and then the immune system or a second mechanism like maybe a therapeutic vaccine, let's say, would be able to destroy those cells. So that's a possibility. That would be like a two-pronged attack necessary usually to destroy these cells. So they've actually gotten some decent animal model responses, but they are still waiting to get kind of good studies in humans. So this is kind of a potential area for further study. Also, PD-1 is like a checkpoint inhibitor. So basically, or is part of the immune checkpoint. So this kind of keeps HIV quiescent as well. And PD-1, kind of inhibiting PD-1 will kind of take it out back into the cell cycle, wake up HIV, and allow us to target it in different ways. So this is kind of a similar attack to the latency reversing agents. And PD-1 also kind of has the ability to downregulate CCR5 sometimes as well. So that's kind of a second way in which it could be effective. There's also some gene editing studies that have been interesting. So they basically take the patient's T cells out and then reinfuse them back in after modifying the HIV-infected cells. Actually, they modify the HIV uninfected cells, I should say. So what they do is they take the CD4 cells out. They use this thing called a zinc finger endonuclease, and so it's basically an enzyme that cuts at very specific places and then is able to disrupt the CCR5 genes. They take those HIV uninfected cells and then reinfuse them back into the patient. And then the patient is getting antiretroviral therapy during this whole time. And then basically the idea is that they're going to boost up the T cell count of the patient, and then they'll have more uninfected cells than infected cells at some point if they keep doing this. And then, so they actually looked in this study, also published in the New England Journal, at 12 HIV-positive patients. 6 of them actually interrupted their antiretroviral treatment after getting this reinfusion of autologous cells. And then important to note is that not all the cells were able to be modified by this enzyme, so there's still more work to be done about that. Only up to about 28% of the cells were modified, so that some of the cells reinfused back in were still able to be infected by HIV. But the results here, and this was really just a safety study, so they were just looking at the safety, not so much the efficacy of it. But you could see that these are the total CD4 cells here in black, unmodified in blue, and then the modified cells in red. And these were assessed through flow cytometry. Basically, the 6 patients that underwent the treatment interruption, they basically had a pretty steep decrease in their total and unmodified CD4 cells. And then in this very small proportion of cells that made up their CCR5 modified cells, these actually remain relatively stable over time. As you can see, the red line is pretty flat. So they thought basically the native CD4 cells, they're getting infected and killed at greater numbers, even though that most of these patients had undetectable viral loads. So there was still some HIV, destroying the cells. And then I would say during the treatment interruption, they had rebounded virus, and then they had some more decline, very faster decline in the viral load, in their T cell counts. So over time, these kind of modified cells were more stable than the native cells. And more protected. So if they did this multiple times, there's a possibility that they would have some success. So there's still some work to be done there. Just a quick note about therapeutic vaccines. A lot of the vaccine trials that were done were with weekly immunogenic vaccines, and they're kind of looking at more immunogenic things like CMV-based vaccines, stronger adenovirus vectors. They used to use weaker adenovirus and canary pox vectors. Those actually were not successful at all. But some of these vaccine trials will kind of keep build on the replicative nature of CMV to kind of produce kind of consistent immunogenicity. And maybe these vaccines will be more effective in combination with one of these other strategies, like a latency reversing agent or something. So these probably on their own won't be successful, but will need to be combined with a different mechanism. Broadly neutralizing antibodies, that's another area of great interest. So basically these are monoclonal antibodies against HIV-infected cells. There's a particular one called PG121 that's been pretty successful in animal models and has caused just rapid suppression of HIV RNA, proviral DNA in blood, and lymphatic and GI mucosal HIV. But more work needs to be done. These are mostly in animal models so far. There's a question of whether these will reach all reservoir cells or not, so that remains to be determined. And maybe a strategy of combining these with latency reversing agents will be needed. Lastly, I'd like to talk about a really interesting to me study that was done at Albert Einstein by Dr. Dedekova, and this is a radioimmunotherapy. So basically they take these kind of monoclonal antibodies and conjugate radioactive isotopes to them. And they use radioactive bismuths in the study. And it has a half-life of about 46 minutes or so. And these are alpha particle emitters, so these are not really things that are going to cause you cancer. They're short-lived, but they're basically seeking out HIV kind of like a smart bomb there and destroying it with a short burst of radiation when they get there. So they've done some studies in mice, and they've eliminated, basically implanted HIV human lymphocytes in the mice. So obviously a mouse model for HIV doesn't exist, so they had to figure out a way to get the cells in there, and then they were targeted. But they've tried this in about 15 humans as well, and they achieved a 95% viral load reduction in about 13 out of 15, and complete elimination of 11 out of 15. So kind of a little pilot study was fairly successful. And then they did kind of like a model in vitro study to see if it kind of crossed the blood-brain barrier to get to that anatomical reservoir, and it did. But they haven't really shown this in humans yet, so more needs to be done with that. So in conclusion, I talked a little bit about the HIV reservoirs, how they're composed of the memory CD4 cells, and the importance of really attacking them early if possible, and shifting that reservoir from a longer-lived to a shorter-lived reservoir. And then we do need better ways to measure the reservoir to make sure that we are curing people, and when is it safe to take them off antiretroviral therapy after we think that we've cured them. And then almost certainly there will need to be a combination of approaches, so 2 or 3 of these methods in conjunction to actually achieve a long-lasting cure in most people with HIV. And here are my references, and I will take your questions at this time.